Okay, hello everyone. We are again at coffee breaks with TRANSFAC. My name is Alexander Kell, and so I'm from TRANSFAC team, and I'm glad to see you back. Today we have a very dense program. We are going to talk about very important things about ATAC-seq, ChIP-seq, CUT RUN, and generally about enhancers. And okay, so let's start. I will, so the program is the following. I will show a couple of slides and just to explain to you what is the context of all this, and then we'll go directly online to see how our tools, the TRANSFAC associated tools, can help you to understand all these kind of data. Let me start. Okay, so that's a very old picture, but nothing changes in the world. Well, a lot of changes happen because we get new knowledge, but generally, you know, there's a gene, and there is a coding region of a gene, and there is a start of transcription which regulate the expression of the gene, and then the regions around the start of transcription are called promoters, actually. Which have some core promoter elements believed to be very important. And then there are some other modules, maybe some distant modules, or quite proximal to the promoter, which contain binding sites for transcription factors. And those transcription factors binding to their target sites, and they regulate activity of the genes. And so that's a general, concept. But then when people started to analyze the composition of, of nucleosomes and basically this DNA sequence, DNA and environment, I would say, around this start of transcription and also around enhancers, they realized that a lot is happening with nucleosomes. So basically, for the transcription to start, you need the chromatin to be opened, and for that, these proteins, nucleosome, which build the nucleosomes, they changes. They got various types of modifications, and they were all studied. Maybe not all, but a lot of them were studied, and then using different types of techniques we are going to talk today. People found that these methylations or acetylations of different positions of of the nucleosome components of histones, they are actually correlate with different types of open chromatin. For instance, if you observe, for instance, trimethylation of histone 3 in this position, then you actually see this signal around the promoter, and a little bit of that signal around enhancer regions. Whereas if you look at, for instance, acetylation of K27 of the same histone, then you observe more signal in around enhancers. Well, for instance, this kind of trimethylation of K36, it's very specific for promoter areas, and it actually even changes towards a particular region in promoter, and so forth. You see a lot of those methylations, which can give you an idea about what kind of opening happenings in the chromatin. All right. So, but those kind of things, they just point us to the important areas in chromatin which would regulate transcription, but we don't know what is exactly happening there. So, and that's just the first step to open the chromatin. But then you have to know which transcription factors are going to bind there and start regulating your genes. And for that, of course, there are various methods to analyze those transcription factor binding. And the first of all, we are talking about ChIP-seq, when we have antibody towards toward that transcription factor. That's known technique, and then by cross-linking of that of that complex to the DNA with a special chemical component, then basically you find the places where this transcription factor bound to DNA, and then after several steps of experimental techniques to clean up this complex and to to get out the DNA fragments where those transcription factors are bound. And then sequencing those DNA fragments, you can understand where those transcription factors are actually bound to DNA in different situations of the cell, in different types of cells, in different interactions of the cells with other cells, and all kind of conditions. All right, but for that, you have to know which transcription factor you are interested. So you have to have this antibody to that transcription factor, and basically then you know where it binds. But there is another technique, a number of techniques called ATAC-seq, where you actually don't know which transcription factor is acting there. You're just interested in open chromatin. And those techniques, they are based on basically cutting DNA when it's not closed by the chromatin, like what you see on this picture, where basically there is a, you see the picture of nucleosomes, like these strange round symbols. And then you see they put the transposase TN5, which cuts the region of open chromatin in fragments. And basically, when the region is not closed, then those, automatic changes. Then you see that you get those fragments, in DNA, and then you get those fragments. You can sequence them, and you can map them to the human genome, and then you see where those fragments are aligned. And with that alignment, you can basically understand where the open chromatin is. Of course, sometimes if you're lucky, you can even see the position of transcription factors sitting on that place, because those transcription factors, they also closing the region, where they're sitting on, and that's why you will get kind of valley in that distribution of fragments when you sequence them. But then, actually, if you're lucky, if your sequencing technique is going very in depth, and you have a lot of reads, then you may observe those valleys. But basically, most of the cases, you just see the peaks, and it's peaks of open chromatin. And then there is another technique which appeared quite recently, which is called CUT RUN, and it's actually based on the modern techniques of cutting DNA, which are very popular now. And I will not going into into biochemical details how it is happening, but then actually it's more or less similar thing like with ATAC-seq or with a ChIP-seq, when using antibodies which are attached to a special enzyme which cuts DNA. You actually cut the DNA in the places where the transcription factor is binding, or in the places of your interest. For instance, if your antibody is not towards a transcription factor, but to a particular methylation status or acetylation status of nucleosomes, then you also can cut those regions and see whether they open or whether they in particular status of methylation or acetylation. So this technique is supposed to be very good and actually very effective. If you read about this in the literature, you see that it's claimed to be very specific and sensitive. Actually, not specific, but rather sensitive and specific. And the reason is that when you do cut and run, this is actually two pictures of the same region, which is done using ChIP-seq technique towards this trimethylation of K27 of histone 3, and using the cut and run technique. And you see the same regions, but much more pronounced. Same peaks, more or less, but much more pronounced. And in the case of ChIP-seq, you see a lot of background noise. And the reason why is that happening is that actually when the ChIP-seq technique is based on cross-linking with DNA, chemical cross-linking, and that gives a lot of background noise and false positives, basically, whereas cut and run is much more clean. So that's why actually it's also much cheaper, because you can get the same quality of peaks by doing much less reads, by sequencing much less, and then this gets much more effective. All right, so but at the end, it's very similar. And at the end, you actually get kind of peaks, as I explained, either by doing ATAC-seq when you are just focused on open chromatin, or by doing ChIP-seq or CUT RUN, which are focused on particular either a transcription factor or methylation or acetylation of nucleosome to understand which areas of genome are active in the case of being active as a promoter or being active as an enhancer. All right, but once you have those sequencing reads, and once you map them on genome, you are here in the area of advanced analysis, and that's the place where we are going to talk today. So I'm not going to talk about all these pre-processing steps, which you can find elsewhere. So basically, there are plenty of tools which are doing that. But once you have your reads mapped to genome, then the real science begins when you actually have to understand why those peaks are there, what they are doing, which transcription factors are actually binding to that place. And then if you find those transcription factors, maybe you can understand biology of that in general. And in case of you are studying some disease, you may be also interested in to find drug targets, biomarkers. So that's the kind of order where you want to go. All right. So and we are going to talk today about peak calling a little bit, but mainly about how to analyze those peaks. All right. So if you talk about peak calling, this is a picture from the same review paper. It's not my picture. Just to impress you a little bit, so to see how many programs are there to do the peak calling. And but generally, there are three types of algorithms for peak calling. One first, which is most popular, is based on counts. So when you read, when you sequence your reads, then each read is one count, and then each region of DNA where you observe those reads mapped to DNA, and we'll see it today, you can count how many reads are mapped to that region. And then basically on that, those regions where you have a lot of counts, they are kind of more probable for binding of that transcription factor or probable for being opened as a chromatin. And then you simply count the number of those counts, those reads. Another type of peak calling algorithms are actually based on shape. So if you put all the reads along the DNA, and again, you count the number of reads, then you can observe kind of a shape of that number. And then based on how shape behave, you can specify those areas which have a very particular type of shape. And then you call them as a peak. And why you actually generally doing this, although the original data is really reads, so they told you where potentially the transcription factor was sitting, but still you need this peak calling. It's a kind of artificial, but also ensuring that you are throwing away false positives, so kind of a background noise. So you're really focusing on those areas of DNA which have a maximum number of probability to be really bound. But you have to understand that's all quite artificial. And that's why different algorithms may give you different number of peaks, different regions under the peak. And then you have to take this into account. So today we will talk about one algorithm, which is MAX, and we'll use it also in our tools. All right. So there's concerning the CUT and run, there is as you can see again, these peaks which you observed by CUT and run. They are it's a very sensitive technique. That's why there is a special tool which is there to find those peaks for cut and run. And that tool is called this SEACR, actually. We don't have this tool in our platform, but you can find it also in the internet. Although these peaks can be found by the standard tools like MAX, and that's what you will see today. All right. So I think now we will just one last slide about basically how we are going to analyze those peaks. And we are going to find transcription factor binding sites in those peaks. And basically, we are talking about enrichment of those binding sites. And just to remind you a little bit, so how we are actually searching for those binding sites, you will see it in my previous coffee breaks, more in details. But basically, in TRANSFAC, we have those. It's a big library of positional weight matrices, which is you can see here. And basically, TRANSFAC is one of the biggest collection of positional weight matrices. Now we have more than 10,000 positional weight matrices collected in this database. And then for each matrix, the algorithm, which we'll see today, basically searches in the region of your interest. It could be the promoter of a gene or a peak, which you get from ChIP-seq or ATAC-seq or cut and run technique. And then you search for, you predict potential binding sites for these transcription factors. And for those predictions, you need a cutoff for the score. So the score of that binding of this matrix, we call it a site score, is varying from 0 to 1. 1 is the maximum score. And if you have a cutoff, say 0.7, it means you will claim all sites which have a score higher than 0.7 as a predicted sites. And then our algorithm will define actually which sites are really enriched and which sites are not enriched. All right, so that's a little bit of introduction. All right, so okay, so we'll stop here for the slides. And let me go directly to our platform and show to you basically how we going to analyze this data. So what you see in front of you, it's a geneXplain platform, which is used here to analyze various types of data. And as you can see here, there's a little button here for ChIP-seq data, which involve number of tools and pipelines to analyze this kind of data. So not only ChIP-seq, as again, I'm saying, although it's called ChIP-seq here, but it's also ATAC-seq, cut and run. So those kind of those kind of techniques, which gives you regions in genome, which are somehow associated with open chromatin or binding of transcription factors, so which associated with gene regulation. All right, so and in the left part of that screen, you see the our project. So I have a lot of projects. So if you have your account in geneXplain platform, you will see only one project, your own project, but you can create new projects here where you can put your data and then use some of those tools to analyze your data. Okay, so what kind of data we are going to see today? So let's start from one example, which is a very simple example. These are the HeLa cells. It's a pretty old data, so 2010, but it's not bad. It's very good data. So these were HeLa cells, and in this paper, researchers, they took antibody to the transcription factor called E2F1. And this is a very important transcription factor, which actually controls the transition from G1 to G1A phase. It's very active in G1S transition period of the cell cycle. That's why these transcription factors are also important for such diseases like cancer and other diseases which are related to cell proliferation. All right, so using ChIP-seq technique, basically, they analyze this data, and here you can even have the BED file of those peaks. But today we are not going to take that, although we can, and we'll see it in a second example, just getting the ready peaks. But today we are going to dive into SRR. So basically, this is an archive where the sequences are stored. So these are actually the fastq sequences which were sequenced on those after doing ChIP-seq. And here you can download those fastq files. So here you see the fastq download, which you can do on your computer. And then you can upload this into your computer, into your system here. So let's just do it right away. So actually I have uploaded it before, but just to show you how to do this, you actually click here or load your sequence, or click on this button, and then you go to your computer, and then you select the file which you want to find, and you see this fastq file. So it uploading into your computer. So actually it takes quite a while. I will not going to show it to you online. So, but you see it's uploading, then it's converting into correct format all automatically. So I will simply drop it now, but I will show you that it's in fact was loaded here, and here there's this fastq file which has a size of 2.7 GB. So it's quite a big file, not really big, but it's kind of standard fastq file. And then the next thing to do with this fastq file is to map it to genome, right? So, and that's what we can do here in this NGS button. So you go to the NGS button, and here in NGS, you can, for instance, use alignment for fastq files with using bowtie 2 or any other aligner of your preference. Well, fastq bowtie is pretty robust, and simply take this fastq file, drag and drop it here. And here you have to select which genome you want to map it to. So I would select the latest genome, which is 38 from human. And well, and then you actually specify your output file. Yeah, let's call it 1. And you start your alignment. Well, alignment takes much longer than just loading this data. But still, you know, it's in fact what you can see here. It uses Galaxy tools, and in fact, just a little bit, those who doesn't know yet. So in this geneXplain platform, we have actually a Galaxy, many Galaxy tools integrated, and it's actually the whole Galaxy platform is naturally integrated here into this platform. So any new Galaxy tool is simply can be copied here and can automatically be invoked from this platform, and also being part of pipelines of that platform. So you see it's using now the Galaxy bowtie from Galaxy to align my sequences to genome. So again, I will not going to do it right now. I will simply show the final results. So it generated this BOM file, and to see it, let's open it on our genome browser. And here I click on databases, basically to show you what kind of databases we have integrated. We have inside this platform. For instance, for human, we have this latest ensemble build, and we can open it in our internal genome browser. And in here, we can simply drag and drop the BOM file to see where our reads. So okay, so you see these little green dots. Maybe let's increase this a little bit. So you see those reads already, right? And here above you see the genes. So these are the little reads. If you zoom it a bit more, you see some of them have differences from the reference genome, which is natural, either because of most of them actually through the sequencing errors, but some actually correspond to real variations in this cell line, which were analyzed here in HeLa cell line versus human genome. So we see these reads, but now we need to find peaks of those reads. So to find those regions where those reads actually agglomerate in some bigger peaks. And for that, we need to analyze, to apply this peak calling algorithm. And now we go back to this ChIP-seq region here, and we go to peak calling, and there are a number of peak caller algorithms. And the one which we are going to use it today is MAX. And MAX is has quite a number of parameters. So, and then you put here the bound file, and you can keep these parameters as they are, or you can, if you know how to specify parameters. For instance, I want to have more peaks. By default, this MAX is actually set up to find only the biggest peaks in genome. And for many reasons, I can explain it more in details if you like, if you ask. The biggest peaks may be not so interesting, in fact, and you have to look also to smaller peaks when they actually cover the regions which are regulating gene expression. So that's why this enrichment ratio can be put to smaller values, for instance, 10 or even smaller. And then you will find not only peaks, those which are enriched. Enrichment of a peak is actually enriched. The number of reads in the peaks is higher, significantly higher than in random case. And this random control case you can actually get and put here into a peak caller algorithm if you have a control experiment by itself. If you don't have control experiment, but just one data, then this background is computationally calculated in this MAX algorithm. And then you still find those peaks which are above a predicted computational noise. Enrichment, right? And this enrichment is set here quite high, and you can put it lower. So basically then you run it. So it also takes quite some time. So I did run it, and this is the result of my run of this MAX algorithm. And here you see we found 15,148 peaks around this maximum number of reads in the genome. So you can see those peaks if you simply drag and drop in here. Basically, you can zoom it out. You see this is a region with many reads, and it covers by this peak. Actually, if you open these peaks as a table with a right click, you can see also this enrichment score, fault enrichment. And for instance, you can also sort it by this fault enrichment, and you see this peak, for instance, of the size 182. It has a maximum enrichment, so it has a lot of reads. And you have other enrichment here, and here in total we have 15,000 peaks. But just to let's have a look at those peaks which have a maximum enrichment. Although I told you that maybe often they are not so interested, but sometimes you're really interested what happening in those peaks which have a maximum enrichment. So for that you can do the filtering here with a filter. You can say above threshold, and your fault enrichment should be higher than, for instance, 10 or maybe 25, and then you apply it here, and then you find those peaks. So actually, I did those filtering, which takes some time as well, and here are the filtered peak, so which are higher than 25. So we can jump to one of those peaks. For instance, this is a peak which is higher than 25, and indeed you see quite a lot of reads here. There is another peak, so you can jump from one peak to another and see where they are really the biggest peaks in genome. And you can see how many peaks we actually found, which with a high enrichment. So if you click here, you see how many peaks here. So we just let me right. So here we have peaks. So we have actually 242 peaks here. So it's quite a small number, but these are very big peaks. So what we want to do is then we want to find enrichment of transcription factor binding sites in those peaks. Why? Because these are peaks where E2F1 is supposed to bind. So, and it's kind of reasonable to expect that the E2F motif should be enriched in those peaks. All right. So how to do that? Again, we go back to our ChIP-seq list of pipelines and go to the analyze ChIP-seq peaks. And here we have this identify enrich TFBS in ChIP-seq peaks. All right, using TRANSFAC. What else? So we have TRANSFAC, and then you take those peaks, you put them here. You have to select the species here. It's a human. Well, actually, by default, it doesn't know from which species these peaks are coming. So you have to tell it explicitly. And then you see below here the whole pipeline. I will explain it a little bit later. But first, we have to define a profile. What is a profile? Profile is a list of positional weight matrices which we are going to use now to analyze those peaks. Here, by default, there is a very small subset of those PVMs from TRANSFAC, which is called non-redundant set. It's very small, just for very quick and first run. But we want to analyze all kind of motifs there. So for that, we go to TRANSFAC here in the database button, and here in TRANSFAC in the data, among the profiles, you see all kind of profiles here for different types of, for instance, for vertebrates, for plants, for other big, species or big classes of organisms. And here, let's select this one, which is called vertebrate human P001. It's maybe a strange name, but just quickly to explain it to you what it is, if you click on it, make a double click. So it contains 8,000, a bit more than 8,000 PVMs for transcription factors, which are working in vertebrate organisms. So including many different species. Of course, we were building it from various particular organisms, from human, mouse, rat, from zebrafish, for instance. But as you know, these transcription factors, when they're binding to their motif, they're quite conserved. This binding motif is quite conserved among big classes of organisms. And we can unify those PVM to be applied to any kind of vertebrate genomes. Human means that those cutoffs, which I was talking before, so which you see here, each PVM has a cutoff. So which means we consider binding sites, predicted binding sites, only if the score of that binding site above these cutoffs. And how those cutoffs were calculated. So we actually calculate such a cutoff that in random case, in a random place of human genome, those sites would be found in average around one site per 1,000 nucleotides. So these are theoretical estimated p-values of those sites in a sequence with the same composition of nucleotides and dinucleotides as in human genome, and that sets for us the cutoffs. All right, so that's a little bit of introduction, but we want to use this profile here. So we take a drag and drop in here, and basically we are ready to start. We name this, name the output in different, and we run the workflow. So as you can see, this workflow started to run, and it has many steps. So first, it's prepares this track, this peak file into a proper format. Then it run the site search in the regions, in the peaks, in this 242 peaks. We selected these filtered peaks. You remember, not all 15,000, but only those which have the highest fold. And then it will do, it first will search for binding sites inside those 242 peaks, and then it will search for the same binding sites in the background regions of genome in the control. And then it will compare the frequency of the binding sites found inside the peaks to the frequency of binding sites for the same factors in the whole genome or in regulated regions of genomes. Actually, as a control, we use regulated regions of genomes, we know. All right, so, and by doing it, it also actually optimizing the cutoffs. It's not only taking those cutoffs which we theoretically predicted, it actually tried to optimize those cutoffs for each matrix in such a way that this ratio between we call it the yes set and the no set of fragments will be maximum. All right, so I will stop it now. It's quite a long process to do that. So we, of course, I did this run before, and we will look at this output. This is a folder here as an output, and basically what you can see by clicking this site search summary, you see this list of motifs which found to be significantly enriched in that areas. And you see there are quite a number of them, 3,000. Out of 8,000, 3,000 motifs actually found to be enriched. So these regions are really, really different. All right, so, you see many different motifs. And actually, now you see the motif for E to F6 somewhere among the top motifs. But you see they are actually sorted by yes to no ratio, by the frequency difference. And let's sort it by the p-value. So to see the actually those motifs which have the lowest p-value, and bingo, we have this E to F motifs as a top motif found here. All right, but why it's not E to F1, but E to F2? And that's an interesting question. That's actually goes towards what kind of factors each motif can recognize. And because of, again, the DNA binding domain of many factors are quite conservative, E2F1, E2F2, all family of E2F factors actually essentially binding to very similar motifs. And independent how you build this motif, from which sub group from a family, you have to assume that all members of that family can bind to the sites found by this motif. All right, so we have this motif, and we can actually visualize this motif where it is in fact binding, and you see it like this. So here we have this 242 motif, 242 regions, these peaks, and inside each peak, some peaks are actually cover some areas between the genes. Some have they actually cover some exons of some genes. But it doesn't matter. Actually, these are areas where E2F was bound. And here, each of that little arrow is one predicted site for E2F transcription factor. Actually, you can see it even better on Genome browser. We return here. And then if you simply drag and drop this track here, then you see where your sites found. So for instance, here in this peak, which you see here by many reads, we found here two sites for E2F. They're often these sites are quite, symmetric, and they found in both directions. And if you look at each of that site more in detail, so for instance, here you see this site has a score of 0.78, and this site has 0.79. But basically, it's a CG sort of repeat, which is found here. And if you zoom it even further, you can see the CG dinucleotides found in that area, which also, as you know, prone to be methylated in DNA. So that's a really region can be a lot of epigenetics can play around with these regions. So, all right, so that's the first example how we find enrichment in one transcription factor from simple ChIP-seq data. So if you have any question in this moment, just go ahead. Let me come to a second example. Actually, now let's talk about a more complex setup. So for that, I will go to this example. This is another pretty recent paper from March 2024, where what these authors of this paper did, they actually are studying B-cell lymphoma, and they found out that it's a quite known fact that the transcription factor called FOXO1 is very important for this lymphoma. And they were interested to knock out this transcription factor. And they did it very smartly. So they actually made a construct which does this knockout conditionally. So which means very rapidly you can degrade FOXO1 in a living cell, and then you will observe what's happening. And actually, they clearly observed that those cells start dying, so they go into apoptosis, which is a kind of a clear reason. So since this tumor, this ever-growing B cells, they are dependent on FOXO1. When you degrade this FOXO1, kind of if you would have a drug which would repress this FOXO1, you will kill those cells, and that's what people want. Of course, it's difficult to repress the transcription factor, which is inside the cell, and we'll talk about a little bit later what to do in such a situation. But basically, when you do it experimentally, just to degrade it, you see the apoptosis. But of course, they were interested to know the mechanism, molecular mechanism, exactly where these FOXO1 were sitting, in which areas of genome. And they did two experiments. They did ChIP-seq and CUT RUN, and actually they also did transcriptomics, and they did it in time manner. So they did it in T0, in time 0, when the cell is like a normal cell, normal B cell, a lymphoma cell, so these tumor cells. And then then they did an inhibition of FOXO1, and they measure again after 6 hours. They actually made several measurements, but for us, interesting to see what's happening in 6 hours. And they did it using ChIP-seq towards this acetylation mark, which you may remember. This is associated with enhancers. But actually, today for us is interesting more towards promoters. And they did it with the CUT RUN, which they were towards the trimethylation, which is a marker of active promoters. All right. So, and then they also did transcriptomics in a separate data. I will show it in a moment. And then actually, we know now then the areas where this trimethylation was enriched. So it means active promoters. And also we will see which genes are up or down regulated upon this inhibition of FOXO1. So with doing that, we want to analyze those areas from a point of view, not just binding, but also from a point of view of the effect, how this binding of FOXO1 to those regions affects the regulation of genes. So in our previous example, we were not considering any effects, just E2F is binding, and that's why maybe the largest peaks in the genome, even irrelevant from a gene where they are basically positioned in genome, just collect the transcription factor binding. But here we are interested in lower peaks, which are maybe located in a near start of transcription of genes, and the binding of these factors actually influencing expression of those genes. And one concept I want to again repeat here, that transcription factors not working alone. They are actually working in composition, and we have in our tools a system which is based on genetic algorithm, and it searches for site composition. So for instance, here in the center, we have a binding site for OXO1, for instance, but this binding of OXO1 is associated with a binding of other factors, which are in synergy with this transcription factor regulate the gene. So like in this picture, you see, for instance, here on this button in this part, so you see this when those composition of factors binding to the region and binding to each other, they form a complex, which is some people call enhanceosome. And after this formation of that complex, they activate the gene. So that's why we are interested to find those compositions. All right. So let's go back to our tool. And now we are switching to another tool. But before doing that, I have a question. The question is, how much time will an analysis take from data loading, for instance, 2 GB to receive the result? So in this pipeline, which I showed to you, okay, so alignment on our server of this 2 GB took around 20 minutes. Then the peak calling, another 20 minutes, and the search of the motifs was another 20 minutes. So approximately in 1 hour, you will get such a result, right? All right. But now we are going into a different tool, which is called Genome Enhancer. And I will show you the trick. Here in the right upper corner, you can switch the tools. So now we are switching to Genome Enhancer, and we go to Start page, and you see a different interface. And that's much simpler interface towards compared to what I showed just before, where you have to do things manually, run tools, see results, run another tool. Here, this Genome Enhancer is one big pipeline for multi-omics analysis. And here we have multi-omics. We have CUT RUN and transcriptomics. If you want, we can add more other omics, but at least here we want to analyze 2 omics together. And then this pipeline will do the analysis, everything automatically by combining these 2 omics data. And I will show, while it's working, I will show to you a bit of what is actually doing. But actually, the first step is to create your project or open existing project. So we have this project, and here you have to load your data, kind of put them drag and drop, sorry, in this area. And I will show to you the data which we're going to drag and drop. Basically, first thing is this expression data. This is a file which I downloaded from Gene Expression Omnibus of that experiment, which has all the genes, how many, like 25,000 or something. Here's the ensemble, the gene symbol, and here you have these various expression values of those genes. Like you see this row counts, like 63 read counts here for this gene. Maybe I make it bigger a bit. And then there's a famous FPKM values, so it's kind of averaged and normalized values towards the length of the gene. And then in which conditions. So here you see T0. This is in T0, right? Here again in T0, I guess, FPKM again in T0, but then you have it also in T6, right? So let's open it in T6. So we have values in T0 and T6 in replicas. So we have this expression data. And this second file is this interval file, which gives you actually the peaks. So you see, these are the peaks of CUT and RUN on this trimethylation of the histone. And here you have a chromosome position, and there are other values like again enrichment, log-fold change for this peak. All right, and we have a number of peaks. So we want to put these two files into our tool. Yeah, let's return to tool. So what we do is simply drag and drop it here like this, and then like this. Like you see, it's automatically uploading. Then we import it, and it's detecting the format of that file. And actually, it found out that actually it's a transcriptomics, obviously, and it defines ensemble, so we can import it, and then we can have a look at this file. And it's also found out that it's epigenomic. But actually, you have to know what kind of build it is. And from the paper, know that when they were finding those peaks, they actually were building it on previous genome, not on HD38, but HD19. So that's why I select this HD19. And it's epigenomic, and we also importing it. So while it's importing, let me show you a bit of what is going to happen here. I will open this slide. So actually, what's happening here, we have genes which are differentially expressed upon treatment, right? So now we compare the control, so T0 before treatment and T6 after treatment, after this inhibition of FOXO1. So first, what the program will do, it will compare these FPKM counts, and it will find differentially expressed genes. So we'll get a list of genes which are differentially expressed, which are upregulated or downregulated after such treatment. So and then what next? So we'll basically analyze transcriptomics. Next, it will actually start looking at the promoters of those genes. And it will take epigenomics data, these CUT RUN peaks, and will intersect it with the promoter areas, with 5' areas of those genes, and find those regions where we have a 5' area of upregulated gene, and we see the peak of that FOXO1, not FOXO1. We have a peak actually CUT RUN, which is actually trimethylation of the chromatin, so which means it's active promoter areas. All right, and then next step, it will start analyzing the motifs in that area, and that will be doing exactly what we did before, so searching for enrichment of all kind of motifs there. And next, it will actually look for combinations of those motifs, not just individual motifs, but combinations of motifs. And then from that, it will speculate what kind of transcription factors are working here, and hopefully we'll find FOXO1, but maybe not. So those factors which are actually working on that in that situation. And then it will ask a next question, how those transcription factors are actually regulated from upstream signaling. And the goal of that, so we will not have proteomics to drive that search, but without proteomics, it can also work just using the standard knowledge. And finally, it will find, we call it master regulators. All right, so that's how it will work. So that, let me open this again. So you see this, these two data were already uploaded and imported. And what we're going to do, we have to drop them here in this gray box, this one and this one. And then we go next. And we have here experiment and control. So basically, these peaks were done just on experiment. So on actually the non-treated cells, right? So, and then the experiment, so T6, FPKM T6 are here. So these ones, this one, and this one. And the control, so, no, wait, these are T6, right? So we want to actually compare the non-treated status to the treated status. So we put this in a control. Well, let's rename it, not to mix it up. So this is a T6. And this is a T0. Why we were comparing this way? Because we are interested to know where FOXO1 was sitting before it was destroyed, right? It was sitting in T0. In T6, it was destroyed. So then we put here the expression values of, it's T0 here, T6. This is a T0. And someone more, I think this one, T6, T0. All right, I think this way. All right, so then what is next? We have to specify the disease, which was a was it cell leukemia, lymphoma? Yeah, B-cell lymphoma. And then simply we compare T0 to T6, and that's it. So basically we are ready to start the analysis, and then the analysis initiated, and this one will take longer. It will take maybe 3-4 hours, because it will not only do all these kind of comparisons, but it will try to find the site composition, which is quite an intensive work. It takes it using genetic algorithm. So we are not going to wait for that. So let's maybe stop it, which you can in fact interrupt and run it again later. But what we will do, we will actually look at the results of that which I did before. So I did it in the last coffee break, actually, which was about the same thing. And then I will see the reports. So, and that's exactly the report which we are going to look at now. So this is a final report of such analysis. You see, you don't need to do many clicks. You just wait 3 hours and it's done. And first, it actually checks what transcription factors are enriched in combinations in those peaks. And voila, we have FOXO1. So it found out of 10,000 motifs, well, 8,000 motifs, it's indeed found FOXO1 here enriched. And then it also found other factors which can work, probably working together with FOXO1 in that environment. And it found the master regulator, which is SEACR, and we'll see a bit of that. So you see also this step of starting the analysis. Actually, it's doing all these kind of steps, like identifying the differentially expressed genes, upregulated, downregulated, building these heat maps of them, also classifying those genes according to different pathways, gene ontologies. So we are not now focusing on that. So for us, what is important is this combination. So as you can see, it found FOXO1 binding sites together with CRL, twist, and also nearby there is another complex of factors which will include SMART and other factors. So, and this combination differentiate those regions in the promoters of upregulated genes from other regions, actually from promoters of non-changed genes. So the red bars are this kind of a cumulative score of those sites together in the promoters in the areas of activity of the cut and run activity of those upregulated gene promoters. So we really see quite a significant discrimination, and the AUC value area under curve is very high. So this gives us really good evidence that these are indeed those factors which are playing here an important role. And then what it's doing next, as I said, it actually builds such a network upstream from those factors. Let's see it in more details. So let's have a look. You see that these are the some of the genes which are upregulated. These are the transcription factors which are regulating activity of those genes in their combinations. And these are the upstream network, which ends at the top by so-called master regulators. So among them, this erzine gene, which was found picked up as one of the most important target, that's indeed the true. But among others, we see also interleukin-4 receptor, IgB is its antigen is very important for B-cell leukemia, and we see also chemokine CRC6CR4 found as a master regulators in this analysis. Well, it's actually even doing more, which basically maybe not is so important for the discussion today, but actually it goes down towards predicting which drugs, either known drugs, clinically validated or even FDA approved against large B-cell lymphoma, toward those targets which we found here as a master regulators, or those drugs which are already known, or it actually made an attempt to predict, to repurpose drug, to predict those drugs which may be also useful here in this situation. All right, but that's a little bit of towards the more kind of clinical application of that. In fact, again, we know FOXO1 is a transcription factor, very important for this kind of leukemia, but it's difficult to inhibit it directly. By building this network, we know which key molecules, which master regulators, which are more closer to the cell membrane, like receptors or other kind of extracellular molecules, may play a role in activating through the signal cascade those transcription factors and activating the FOXO1. So this can be our targets for treatment, because through that they will lead the signal towards nucleus and the final effect. All right, so that's kind of a story how to start from simple measurements, what areas are open using CUT RUN, but then we analyze those areas, we predicted which transcription factors are important, and then we can even build a bigger story, like what is a master regulators, what can be the drugs which can be used towards these targets. All right, so that's what I wanted to show today. If you have questions, please go ahead. So we have enough time for your questions. If you are watching this in our YouTube channel, it's a perfect time to put your like. All right, so I have a question. Can we use this analysis in plant science research to develop climate resilient crops? Wow, thank you very much for this question. That's I like that, you have a really vision to save the world. All right. Yes, actually, I already announced. We actually already announced a next coffee break, which will be with a little gap here in October, beginning of October, about synthetic promoters. And among examples which I'm going to show will be also examples of plant promoters, which are quite important area of research. We call that basically as a since this seminar is intelligent design of promoters, and that's a little bit of joke behind, because of course intelligent design is the idea that everything was built by by God, and then here we kind of feel ourselves like gods. So we because actually many functions in plants, in any organisms, are controlled by gene regulation. So those changes in the gene structure, when you have very harsh mutations which changes the gene, maybe knock out the gene or bring a new gene or build a different type of gene, they are very harsh. They are very they change a lot in the system, and it's difficult to develop. But gene regulation is something very tiny. So you can increase the expression of some genes, decrease expression of some genes, and by doing that, you adapt your system to environment, like in your question. So in such a harsh environment, like this pollution, or maybe in this change of climate, very hot seasons, you have to make your plants more stable, or maybe dry resistant. And then basically that could be happened by regulating genes, expression of many genes. And of course, we have in TRANSFAC matrices for plants. Although for plants, it's of course majority of those positional weight matrices came from model plant organisms like Arabidopsis, but also from a few from other plants. And plant biology is very wide, and it's difficult to compare Arabidopsis with other type of plants. But to a certain extent, and there are good examples of those approaches, you can apply those motifs also for analyzing regulatory regions of genes of important crops, for instance. And there are examples of that. So yes, the answer is yes, you can. Thank you for your question. All right, so you have another question. For peak calling of CUT RUN histone reads, is SEACR better than MAX in your experience? All right, so actually, yes, it's better. And we have a plan. We have plans to incorporate SEACR in our tools. It's actually makes smaller peaks. And the point is in our CUT RUN in general, because it's so sensitive, people tend to make less reads compared to, say, ChIP-seq. So which means for the algorithm like MAX, it's not enough peaks to decide about the peaks, not enough reads to decide about peaks. But still, it's make a decision, and generally it creates peaks which are wider. But SEACR is actually making smaller peaks, in my experience. Well, depends on the parameters as well. And that's generally better. So you actually know better where the maximum of the peaks are located. But from a point of view of predicting transcription factor binding sites, it may be not so relevant. Actually, I haven't done direct comparison from the point of view of end result. So I mean, which transcription factors are enriched. And then it's actually a question whether you really have to consider very small peaks or bigger peaks. And there are a number of examples when you see a peak, but the binding sites which you found enriched are nearby. Because the reads is just a read, but read is marking maybe a fragment of DNA which was taken by these experimental conditions, by this cutting of the CUT RUN. And then you sequence those fragments from ends, right? So you may be not reading this fragment to the final end, and where actually the binding site for a transcription factor is located. And especially when you have not enough reads, you may have such situation. Then it means having smaller peaks is not necessarily have to be an advantage from the point of view of finding the enrichment of binding sites. But that all has to be approved. Thank you for your question. Actually, we will do this direct comparison, and maybe next time we can report about results. Okay, so another question. Can we use this analysis for microbial genomes? Well, unfortunately, not. We don't have microbial motifs inside our platform, but actually, it doesn't mean you cannot use it if you have those motifs. Actually, if you have your library of motifs from bacterial genome, And there are a number of databases which have those motifs. For instance, there is a database, Mexican database called RegulonDB. There is another database from Braunschweig here nearby. I forgot the name of it. So it's a collection of bacterial motifs, and you can actually load those libraries of motifs in your project. And then there are a number of tools to create profiles from those motifs, and then you can use those profiles using standard tools in our platform to search for binding sites. For of course, you have to have also the bacterial genome, and for that you can also import this genome directly into the system. And you can look at our videos we have already recorded before, where I showed how to import new genome. Actually, I didn't have a yet video for showing importing a library of motifs of your own. Maybe it's a good idea to do such a video. Yes, you can load your motif library, load your genome, and do this enrichment analysis in the platform. All right. But of course, for bacteria, we know very little about this transcription factor, at least inside the TRANSFAC database. There is no knowledge about transcription factors from bacteria. And that means once you found those enrichments of motifs, that's end. So you found those motifs, and then you have to maybe do the Google search or do literature search to understand what these transcription factors are actually doing. In case of human, vertebrate, or to a certain extent also plants, we have knowledge about those transcription factors in our TRANSFAC database. And also, as you showed today, we have knowledge about how these transcription factors are involved in signal transduction network. So we can do a number of good predictions of what's happening in the cell from point of view of the whole mechanism. For bacteria, it's not yet. We hope to have some time in future when we have enough time, and maybe we get some project related to that to enrich our tools from point of view of bacterial genomes. All right. Okay. More questions? Okay, I got one more question. All right. So what are the limitations of CUT RUN Seq analysis? All right. So I don't get it. So what do you mean on limitations? From the experimental point of view, I cannot answer this question because, as you know, we are just dry people. I have very little knowledge about the experimental limitations. But from point of view of analysis, sequence analysis, all right. So I don't see any limitations. So standard, of course, if you have a very big fastq file, which is very rare in case of CUT RUN, then your bow tie may take a lot of time to align. It may take maybe several days on our server if your file is like huge, like several hundreds of GBs. Then of course this might be a limitation, but it will just run. You have to wait. And of course, if you found a lot of peaks, what I showed today is actually, you remember for E2F, for instance, we found 15,000 peaks, which is not very many. But still, I didn't start analysis of all those peaks together using the site enrichment. Because, if you have so many peaks, the site enrichment not only can be very long, but it also can be actually not very sensitive, because you get peaks actually covering basically the whole genome. Okay, 15,000 is not much, but if you have 100,000 of peaks or something like that, then your enrichment of motifs can be dumped by a lot of noise. That's why I showed also here in my presentation, I made a filtering of those peaks by something. For instance, I filter them by the enrichment. So I'm taking just the highest peaks. Or when I'm analyzing it together with transcriptomics, I also not analyzing all peaks in genome, but I'm analyzing only those peaks which are found to be nearby upregulated genes. It's another type of filtering. And so that's kind of limitation. If you want to run the whole 100,000 peaks, again, it will take a lot of time, and it will not usually bring good results. But generally, there is no other limitations. Okay, so the question was from experimental point of view as compared to ChIP-seq. Again, as I said, I'm not a specialist in these experimental techniques. Please read why I'm telling you today was basically what I read and I was scanning it for different papers about CUT and RUN. And what people are saying, again, it's more sensitive than ChIP-seq. It's easier to run from experimental point of view. So that's what I heard. And it's cheaper. So because you don't need to sequence so deeply compared to ChIP-seq. Otherwise, please check in literature or talk to those people who are actually doing it manually. And we have one project. Where our partners actually are going to run CUT and RUN. I can ask them, of their experience and tell you. So, okay, if no other questions, that's what I wanted to discuss today. And please stay in touch. Look at our announcements. Okay, thank you. Goodbye.